Okay, after the completion of the reversible inhibition cases, now we will go to the irreversible inhibition. Remember, the basic difference between reversible and irreversible inhibition is the formation of a covalent bond in case of irreversible inhibition. Okay. And there is no covalent bond formation in case of reversibles. For irreversible, there is a covalent bond formation. Now, I have already told you that there are two types of reversible irreversible inhibition. One, what is called suicide inhibition, and the other what is called active site directed inhibition. Let me see, before we go on to the suicide, I think we will talk about the active site directed irreversible inhibition. It is abbreviated as ASDII. Okay, now, what happens here? The name suggests something, active site directed. That means, you have a molecule which is having a directional property, which is having a property, and that property is directly utilized to inhibit the enzyme. Basically, I can give an analogy that it is like that if somebody wants to kill another person, so he has a gun and he shoots the person straight away. That is what is the active site? The active site in a person is mostly the heart. If the heart stops, then everything stops. So, heart is the active site. If you consider, then this active site directed irreversible inhibition means you now shoot straight at the active site. Okay. Schematically, how it is shown? It is shown that E plus S, that is the normal, goes to the ES complex. Okay. Then in actually it goes to enzyme product complex, but and then that goes to E plus P. Usually for Michaelis-Menten equation, we generally do not consider this fact, because we assume that that is a very fast dissociation. Okay. In active site directed inhibition, what happens? You have a molecule which is represented as IX. This X is the one which is attached to the inhibitor, that is extremely reactive, and I is the part or the whole assemble is complementary to the active site of the enzyme. Although it looks IX, but the complete inhibitor is IX, not only I. Okay. This is the inhibitor, but the inhibitor already has a very reactive functionality, but it has got again electronic and geometric complementarity with the active site of the enzyme. So, now what happens? This IX will go, because it has got this complementarity. So, it goes and binds to the enzyme at the active site, but at that point, after binding, the enzyme realizes the mistake. It has got lot of reactive functionalities, say like lysine NH2 or thiol cysteine, or weight which comes from threonine or serine. So, lot of reactive functionalities you can think of, or acid functionality as carboxylate. So, this is now is a very reactive X functional group. So, that reacts with with one of the amino acid residues that are present in the enzyme active site. So, this is, and then there is a covalent bond formation between E and X, because you are doing a reaction now, and but this. So, even if I tries to go out, but it is now bound to the enzyme via this X E bond. Okay. So, that is what is active site directed inhibition. That means, here the molecule binds, and it already has a very reactive functionality, and then while it binds, the enzyme amino acid residue, which are extremely say reactive functionality. So, that immediately reacts with X, and in the process E gets tied up covalently with this IX molecule. So, even if this portion is broken now, but your enzyme is now covalently attached to this IX. What are some of the examples? See, some of the examples are like this, that if it is containing a carbon with a with a leaving group attached to another a carbonyl like CO CH2X. Usually these molecules from organic chemistry we know that any leaving group attached to a carbon which is adjacent to a carbonyl, SN2 displacement is very high, rate of SN2 displacement, because this is a very reactive system. So, based on this part COCH2X, you can have different X's. These are all different X's that are given here. So, you can have different reactive functionalities attached to this inhibitor, and the inhibition can take place. Okay. But remember, the enzyme has not done any normal reaction that it was doing to the substrate. That reaction has not taken place. It is just the like a magic bullet, it went in and then it forms a cross link with any of these amino acid residues. What is the a better example? Like if you have I told you that most of these inhibitors are based on systems like this, and this is R. They will be all inhibitors because this is very reactive. Now, if you want to design a particular inhibitor for a particular enzyme, then you have to think of what could be my R. Okay. Now, suppose if we have chymotrypsin in mind, we know chymotrypsin hydrolyzes amide bond where from the carboxy side, where the carboxy belongs to an aromatic amino acid like phenylalanine, tyrosine or tryptophan. Now, see, if you have it was also found by people that chymotrypsin can hydrolyze also esters. It not only hydrolyzes amides, the peptides, but it can hydrolyze esters like the ethyl ester of this, but you have to maintain that factor, that it only recognizes aromatic amino acids. So, in general what happens? You have a system like this, what is required for hydrolysis of by chymotrypsin. You have a system like this, and this is also important that should be in the L configuration. Then phenylalanine, this chymotrypsin, as I told you, you know the mechanism now of chymotrypsin, that it is a serine based enzyme, serine, histidine and aspartate, that triad should be there. But apart from that, it must have some pocket which is a hydrophobic pocket, where phenyl or aromatic groups can be stabilized by stacking interactions or what are called this hydrophobic interactions. Okay. And because of the presence of this pocket which only recognizes aromatic groups, chymotrypsin is specific for aromatic amino acids. For lysine, which hydrolyzes only basic amino acids, like for trypsin, which hydrolyzes only the basic amino acids like lysine or arginine. Why it is so specific? Then this pocket is no longer hydrophobic, because trypsin recognizes groups like say NH3 plus, a lysine amino acid or an arginine here. So, I can just represent by BH plus, because they will be all protonated. So, they go there, they first fit to the enzyme. So, the enzyme must have a trypsin enzyme must be having a site where lot of carboxylates are there. So, that you can make salt bridges, that NH3 plus and CO2 minus. So, there will be lot of salt bridges. So, that is why trypsin recognizes basic amino acids, because of the presence of this salt, because of the presence of a pocket which ensures salt bridge formation or electrostatic interactions, and chymotrypsin has a hydrophobic pocket, but which recognizes only aromatic amino acids. Okay. Now, I want to design an active site directed inhibitor of chymotrypsin. How do I do it? This is the mechanism of mechanism, the charge, that relay process. Na? Carboxylate, that aspartate takes up the hydrogen from the imidazole and that activates this nitrogen as a base, that takes up this hydrogen, and this is supposed to go and attack the carbonyl. But if you do not take the amide here, if you take a CH2Cl instead of taking an ester or an amide, then what happens? Two things that are important. One is, now this serine, if it attacks the carbonyl, you cannot break a carbon-carbon bond, that is difficult to break. Okay. Earlier there was carbon-nitrogen bond, that is one point. But the more important point is, now this imidazole is very close to this CH2Cl, this carbon which is extremely susceptible to nucleophilic attack, it is very close to that. So, now the charge relay will be like this O minus takes the hydrogen, this goes here and the nitrogen is activated. So, instead of taking the proton from serine, because that is now useless, even if it takes the proton there is no productivity, because the serine cannot attack this carbonyl, because there is no meaningful reaction here. So, the nitrogen, instead of taking the hydrogen, it goes and attacks this carbon and makes the carbon chlorine bond. So, what will happen, or what is the result of this? The result of this is that you are forming a carbon nitrogen covalent bond with this molecule. Okay. So, this is a very good example of active site directed irreversible inhibition. Why active site directed? Because your molecule has a system which is recognized by the enzyme. Okay. So, which is recognized by the enzyme. So, the inhibitor goes and binds to the active site. Then the normal cycle wants to take place, but that cannot happen here. What is then because it has got a reactive functionality COCH2Cl, now the imidazole goes directly, acts as a nucleophile and attacks this carbon and forming a carbon nitrogen bond. So, this is the first example is the compound is called tosyl phenyl chloroketone, TPCK, abbreviated as TPCK. This is a very textbook chemistry. This chemistry now has appeared in many textbooks, and if you want to, now you can because you have this knowledge. Now, if I ask that how to now make a irreversible inhibitor, active site directed of course, of trypsin. So, what you do? You now replace this phenylalanine group and put, instead of taking this TPCK, that means tosyl phenyl chloroketone, you take tosyl lysyl as lysine chloroketone. Okay. So, that means that will be called TLCK. TLCK is tosyl, this L stands for lysine and then chloroketone, TLCK. This is an inhibitor for trypsin, and the other one, this one is an inhibitor of chymotrypsin. Okay. Both are active site directed, because both the molecules have reactive functionality, number one. Number two, they also have the structural feature that is necessary, which is recognized by the pocket that is present in the active site. Okay. So, this is active site. The next one is suicide inhibition. This is another interesting, very interesting topic. Here what happens is that the inhibitor looks like very similar to the substrate, and enzyme, what the enzyme can do, because it is very similar to the substrate. So, the inhibitor also goes and binds to the enzyme like the substrate. After binding, what happens? Usually the enzyme wants to do the reaction that is supposed to catalyze. Okay. So, it tries to do the reaction on the inhibitor, and it does that reaction. What is it is doing with the substrate, original substrate? It does the same reaction, and but in the process, okay, now it is no longer I, like it is no longer S, it is now designated as product. So, here the inhibitor is converted into another molecule. but ironically this molecule, what the enzyme is doing, it is doing the same reaction like what it is doing on substrate, but in the process it makes a molecule which is highly reactive. Before reaction it was not that reactive, after reaction it became very reactive, and then as it is so reactive it immediately forms, actually this should be I star, E I star. So, a covalent bond is super reactive, a covalent bond is formed. So, basically I can take an analogy. If you have read the story of Frankenstein, what happens? A scientist wanted to bring life to a dead person. So, he created the life into that dead person, and the dead person wanted ultimately killed the scientist. Okay. So, it is a very similar. The enzyme initially thinks that I is my friend like the substrate. So, the inhibitor goes and enzyme embraces it, okay, with both arms, and then what happens? And then for friends we generally exchange words. Here the enzyme that does the normal reaction, thinking that it is a very friendly molecule, but unfortunately after doing the reaction this molecule becomes an enemy and finally kills the enzyme by forming a covalent bond. So, that is why it says that the enzyme is as if committing suicide, and that is why this is called suicide inhibition. Okay. It is also called mechanism based enzyme inhibition, another name is given there, because the whole thing is depending on a particular mechanism, that how I is converted to I star. So, this is also called mechanism based inhibition, irreversible inhibition, and but the more common name is suicide inhibition. Okay. Now, what is an example? Before we go to this, I told you that enzyme inhibitors are very important from drug design concept, from drug design. Many of our drugs, 60% of the drugs are actually inhibitors of some kind of enzyme or receptor. Now, in case of cholesterol, when we have the disease high cholesterol, I want to modulate the activity of the enzyme, lower the activity of the enzymes which are involved in the biosynthesis of cholesterol. But I do not want to shut off totally that cholesterol should not be produced in my body, because cholesterol has other roles to play. From cholesterol we get the steroid hormones in our body. Okay. Cholesterol also constitutes the that membrane that surrounds our that keeps the cell contents intact. So, you cannot completely shut off cholesterol. Like sugar, if somebody is suffering from diabetes, but you have to maintain the level of the glucose at a certain value. If it goes down, that is also a problem from the normal value. But in some cases, so in those cases a reversible inhibition is better, because you take the drug, it modulates the activity of the enzyme, so that your cholesterol level or sugar level is maintained at the optimum condition, optimum what is expected, and then when the drug is metabolized, you take the next day another pill to keep the concentration of the drug in your body. But the important thing is that you need reversible inhibition. You do not want to completely shut off the cholesterol biosynthetic pathway or the your sugar glucose metabolic pathway. You do not want to completely shut down, but in some cases you need this irreversible inhibition. Remember, reversible inhibition can be reversed, the inhibition can be overcome by adding excess of the substrate, but for irreversible inhibition you are destroying the enzyme. Whatever molecule is attached covalently to the inhibitor, that enzyme you cannot recover its activity, it is already gone, it has formed a covalent bond, active site residue is gone. So, but where it is required? It is required when we want to really kill the enzyme totally, like when we have a bacterial infection, and suppose there is a particular enzyme in the bacteria which is very essential for the bacteria to grow. So, then we target that enzyme in the bacteria, and it will be much better if you have this type of irreversible inhibition, because you want to kill the bacteria. That means you want to kill the enzyme. You want to completely shut off the pathway that the enzyme follows. Okay. So, I will quickly go to the example, again a textbook example. This is not a drug molecule, this is a textbook example that whether how to design a suicide inhibitor. Okay. This was this is a molecule, you know, again chymotrypsin was the model, because chymotrypsin structure is well known, its mechanism is well known, it is available in plenty at a cheap rate. So, all these studies initially were done with chymotrypsin. We know chymotrypsin recognizes aromatic amino acid. So, all these examples are based on phenylalanine, that is the easier one to work. So, phenylalanine with a this is nitrogen, that is the carbonyl side of the phenylalanine. You have the carbonyl as protected with isopropyl, and here instead of the amine CONH, what you have is that this nitrogen is also having another benzyl. This is actually benzyl. We call it phenylalanine because we think it is phenyl derivative of alanine, but you can also think of that this is a benzyl derivative of glycine. So, that also you can think that this is called benzyl glycine, but unfortunately from the very beginning it was called phenylalanine. Okay. So, you have now two CH2 PHs. This has a configuration L configuration, okay, and this nitrogen is attached to a nitrosyl group NO. So, what you have is N nitroso N benzyl phenylalanine derivative, okay, and show it to the enzyme. What was the idea of the scientist who was doing this? The idea is that we know that the enzyme, because it is phenylalanine, the enzyme is having this same catalytic triad, and it will come and attack this, and this bond will be broken. Not this bond, sorry, I am sorry, this is attacking the just one second, I will erase that. Okay. So, again just take the pen from here. Okay. So, what was it supposed to do? The which is supposed to do an attack on the carbonyl and break the carbon nitrogen bond, that what was earlier the peptide bond. Here it is N nitroso N benzyl. Because you have this benzyl group in the correct configuration, there was no problem. So, it goes and it binds to the active site, and then this by this catalytic triad mechanism, this hydrolyzes this carbon nitrogen bond. Okay. Now, what happens to this species? The species will be initially like this, pH CH2N minus initially, and so this is basically nitroso benzyl amine anion. So, that will now take the proton, so to make it NH. So, what you get now in nitroso benzyl amine. Now, these type of functionality in organic chemistry, from organic, if you have read organic chemistry, so because this subject is all about organic chemistry and biology. So, you see now the reactivity of molecules come into play. This NH-N-NO, that means N nitrosoamines are very unstable. What happens? They go to this other tautomeric form, that is called the diazo compound, and this diazo compound is also not very stable. Now, this goes to it releases the this OH, the OH comes out as H water, and this forms this. There are some mistakes here. I think better is you write this way, pH CH2 N triple bond N, and this nitrogen is plus. Earlier structure didn't, this is one resonating structure, but this is not very stable one, because this is not having the octet fulfilled. This is the better structure where all the octets are present, all the atoms are fulfilling the octet, but this is also not stable. Now, this triple bond N can come out as nitrogen, a stable, very stable molecule. So, in the process what you are making a pH CH2 plus, which is nothing but a very reactive electrophile. Any alkyl group with plus will be a very reactive electrophile. So, whatever nucleophilic amino acids are there in the active site, now they can react with this enzyme active site and forms, this is the covalent bond that is formed. So, this is first of all irreversible inhibition, because you are forming a covalent bond with the substrate, but this is with the molecule that it is binding, that is the inhibitor molecule. Number 2 is that this is not active site directed, because there is a particular reaction that the enzyme has done. What is that reaction? The normal reaction. The enzyme hydrolyzes this carbon nitrogen bond, okay, carbonyl carbon nitrogen bond. So, that normal reaction has happened, and ultimately what is created is a monster. This is a monster that ultimately creates a very electrophilic benzylic cation, and the enzyme, whatever nucleophilic amino acids it has, it will now capture this electrophile, and in the process the enzyme should be inhibited. Now, the scientist who made this molecule, he had this in mind. Okay. In research we have many things in mind, but that does not work. Here apparently it was very well thought out proposal. So, he made this molecule, show it to the enzyme, that is chymotrypsin. Unfortunately, he did not observe any inhibition. Why is that? That was the question, that why there was no inhibition, whether there is this reaction failed. No, he found that this reaction was going on. That means, this part was released, and this part was forming the acyl enzyme complex, and finally, this was converted to the CO2H, but it failed to inhibit the enzyme. What happened here? The interesting point, because he was a brilliant scientist, so he realized what is happening here. See here, the part which is attached to the enzyme is the phenylalanine, that benzyl part, the phenylalanine, L-phenylalanine, that pH, that is bound to the enzyme, and the reactive species that was generated CH2, and then you have NH and NO, that is generated, that has no connection with the enzyme. So, by the time all these reactions take place, it just has come out of the enzyme pocket. So, it has gone out of the enzyme, because there was no connection of this with the enzyme. The part which was attached to the enzyme hydrophobic pocket was this phenyl that was attached. So, the other part was free to be released, and by the time all these rearrangement reactions take place, that reactive part is outside the enzyme. If it is outside the enzyme, so it cannot destroy the enzyme. So, this did not work, but as I said, he was a brilliant scientist. So, what he did a slight change. What is this slight? This is the one I was telling, that because this phenyl was being attached to the enzyme hydrophobic pocket, this part is free to escape from the enzyme active site. So, then what he did, he changed the configuration of this carbon. He made it a D-alanine, a D-phenylalanine derivative. Okay. This was L. Remember, amino acids, proteins are all made of L amino acids, and so it is natural that the enzyme will also recognize only L amino acids containing molecules. It will not recognize, or it will not recognize the D. So, that means, this phenyl now cannot bind to this hydrophobic pocket. Why? Because the configuration is D. Earlier this hydrogen was alpha, below the plane, and you can think of this phenyl as above the plane, if you think that, and the active site is somewhere here. So, now when the phenyl is above, the plane, hydrogen is below, the phenyl can go and fit into the active site. But when you take the other configuration, now the hydrogen is up and the phenyl is down. So, it cannot go and bind to this active site. But what happened here? We are all now this benzyl was sitting by the side of this of this alpha carbon. When it was L, because there was perfect match in stereochemistry, so it goes and binds, and this is free to escape. When you made it D, then this molecule binds to this enzyme, but via the phenyl group of the N-nitroso part now. Okay. And you know nitrogen, what is the stereochemistry? Nitrogen can actually flip back and forth. Nitrogen configuration is not a particular one. Whatever is required, it will do that. So, if it requires a up phenyl, so the nitrogen lone pair will be down and the phenyl will be up. So, this phenyl now serves as the it replaces the other phenyl of phenylalanine. So, the N-nitroso linked benzyl group, the aromatic ring now is interacting. Now, what is the effect of that now? Now, this is not free to escape from the active site, because it is the phenyl is bound to this point. So, if it is bound, so exact what will happen now? This phenyl, this is the phenyl of the N-nitroso part. So, that is remaining. So, ultimately this species, so whatever time it takes, it will take, but this is already attached by weak interactions. So, it cannot escape from the enzyme active site. So, this goes out, you make the electrophilic carbon here, and now if there is a nucleophile in surrounding amino acid, then that is going to react with the this benzylic carbon. So, this is the mechanism of this reaction. Now, this is an example of suicide inhibition, because as I said, in suicide inhibition the substrate goes and the inhibitor goes and binds, then the enzyme does the normal reaction. Normal reaction means chymotrypsin is supposed to hydrolyze an amide bond, and that what has happened. But after the hydrolysis, the species that is generated, that is extremely reactive, and because of this switch over L to D, this benzyl is bound to the hydrophobic pocket. That means this reactive species is now bound. So, whatever chemistry that takes place after this, even if it takes some time, that does not matter, because the reactive species is still bound to the enzyme, and then the enzyme nucleophile will attack this benzylic carbon and forms the carbon X bond. So, this is the story of reversible and irreversible inhibition. I think now we have completed this inhibition part. So, next day we will see more design part, because this forms the basis of our drug design, the next module, that is related to whatever we are talking now. So, we have to clarify all doubts about enzyme inhibitions, and then we will go to the nucleic acids once this is over. Thank you very much.